Okay, let's get started. So we're up to the question.
Next class we're just doing a case study. Is it on last classes or both combined?
Okay, so this is called the motor control cases. So it's going to be on cerebellum as well as basal ganglia. Okay, so every section is going to be. So we're done with the first part of cases, but you have to know the information from the first half of the semester that it's additive as we go along. Okay, but with motor control, I'm thinking cerebellum and basal ganglia specifically. Any other questions for where we are now? This is basal ganglia. So what I'd like to do is I'd like to take the first part of the class and go through the basal ganglia and their function and exactly what they are and how they work. There are a couple of different pathways, both the direct and indirect pathway that we're going to look at. And then we're going to look at some videos for patients with basal ganglia problems. And then if we have time at the end, I want to finish up the dysdiadochokinesia that we started with last class, or the cerebellar, so the coordination test. Coordination test.
I'm really good.
What happened?
Somebody made a weird noise.
Okay, you ready? Let's get started. So this is going to be my standard sagittal section coming through here. This just and even though it's a sagittal section, this is going to represent the outside. Actually, it's not even a sagittal section. This is just going to represent looking at the right side of the brain. And here's going to be, what is this thing called again? Do you guys remember way back when we talked about this thing? Yeah, that's going to be my central sulcus. Right? Okay, and thinking about the basal ganglia. And what we had talked about previously is just rostral to the central sulcus. We had this one area that we called M1. Okay, so everybody remember M1, and that's going to be, another way to say that is your precentral gyrus or your primary motor cortex. We talked about that before. And then just rostral to M1, we had, and more dorsal, we had another important area that we called the supplementary motor area. And we talked about the SMA, or the supplementary motor area, was involved in motor planning. Okay, supplementary motor area is involved in motor planning. And then just ventral to SMA, we had another motor planning area that we talked about, and it's called the PMC, or premotor cortex. So just a little bit of review, things we talked about that are involved in motor function previously in the semester. Okay. And we also know that involved here in motor control is going to be this structure that I represent like a peace sign. And this is going to be your thalamus. Okay. And before, talked about different areas of the brain that are going to be important for motor function or motor planning. And one of the areas that we talked about specifically, and I'm just going to write them in here, we'll talk about exactly what they are and what they're composed of. I'm just going to abbreviate them as BG for the basal ganglia. Okay, so today we get to learn all about what the basal ganglia are and what they actually do and how they work, really. If you remember before, what I talked about was information coming from the PMC, and the SMA would come down and ask questions of the basal ganglia, and there was some information going back and forth between the two of them. Okay. And then there's also, today you're going to find out that the basal ganglia talks directly to the thalamus, and that thalamus in turn sends information back up to the PMC, and SMA. And this is going to be all information involved in motor planning. So that when the SMA and the PMC communicate their information back to M1, they're involved in determining whether that upper motor neuron that resides in M1 is going to fire or not, or how much it's going to fire. If you remember, information coming down from M1, so this upper motor neuron sends out an axon, travels all the way down, crosses over, and is involved in activation of skeletal muscle, so voluntary movement. Muscle. Nothing new yet, right? Everybody remember, this is just a quick refresher. Things we talked about that are important for motor planning and movement in general previously. Okay, it's all right here. Now, today what we're going to talk about are the functions of the basal ganglia. So if I have to talk about the basal ganglia itself, there's some functions that you should know. Does anybody know any functions of the basal ganglia? Anybody in P&O exercise? Oh, P&O, sorry. Parkinson's exercise group? Somebody's in Parkinson's exercise. Yeah? Okay, well, Parkinson's going to be one of those things that we talk about today. So one of the first functions that you need to know about the basal ganglia is they're super important for initiation of movement. Okay, initiation of movement. And one basal ganglia disorder that you're going to see this very much in is going to be Parkinson's. People that are in the Parkinson's exercise group have a little bit more of an understanding of this, but I know that you may have seen before in these patients that especially when they go to do something or go to turning is going to be a big one. They're trying to go, but they really have trouble going. Or even sometimes if they want to walk, when they want to, it's the start or the initiation of the movement that's going to be really hard for this patient population. Okay. What we're going to see is Parkinson's is an example of a basal ganglia disorder, and it involves something known as the direct pathway. We're going to talk about what the direct pathway is today. Okay. So another function, second function that you need to know about for the basal ganglia, and that's going to be in the inhibition of unwanted movement. Okay, inhibition of unwanted movement. And what I mean by that is if I'm going to do any kind of motor task or even a fine motor task, for example, if I wanted to brush my teeth, right? I've got a toothbrush in my hand and the rest of my body stays pretty much still. It's just this that's got to be moving, right? I really have a hard time brushing my teeth if I was all over here, right? That would be unwanted movement. So it's going to be the indirect pathway that's involved in inhibiting that unwanted movement. An example of a disease process for the basal ganglia that has a lot of unwanted movement, and we're going to talk about Huntington's disease. Huntington's, and the pathway that's going to be responsible for inhibiting that unwanted movement that we're going to talk about is known as the indirect pathway.
that the same as like Parkinson's patients having tremors, or is that?
A little bit different. So Parkinson's patients' tremors are due to a different pathway, so a different neurotransmitter. We're going to spend our time today focusing on dopamine. I like to keep things simple for the most part, but the tremor is due to acetylcholine. It's a little bit different. So this is more thinking about postural stability for doing tasks. So it's known that even if I just wanted to move my arm, my trunk has to fire first, and that's an example of the suppressing that unwanted movement done by the basal ganglia. The third function of the basal ganglia. So we have initiation of movement. You have inhibition of unwanted movement. And the third thing that the basal ganglia is responsible for is modulation of the amplitude of movement. Does anybody know what I mean by amplitude? So like how big something is, or how small something is, or the size of the movement in general. Okay. And an example of that would be if I wanted to erase just one word up on the board. Okay, I know that to erase one word, I have to be able to control very specifically the amount of movement that I have here. Okay, I just want this one space. But that's a very different thought than if I want to erase everything, right? The amount of movement that I have to erase everything has to be a lot bigger, right? So that's going to be an example of the modulation of movement. Patients with Parkinson's disease do have modulation of movement problems. Everything with patients with Parkinson's is going to be small. Okay, bradykinesia, that means really slow movements. Okay? Their voice is going to be really soft-spoken. Okay, lots of different things that we can think about with Parkinson's patients. So modulation. So 3 different functions of basal ganglia you need to know: initiation of movement, modulation of movement, and inhibition of unwanted movement. Questions about that? All right, let's move on then. The next is, what are the basal ganglia? I've sort of seen bits and pieces of these structures previously when we looked at some of the pathways or some of the tracks, spinal cord before. And we're going to talk about exactly what these structures are. So here, up top, this is going to be a standard frontal section. Hopefully you're used to seeing that. Here's my thalamus. Okay. And in addition to my frontal section of the cortical regions, just underneath, this is going to be a transverse section down here. This is my midbrain. And I know it's my midbrain because I've got my nice little marker, the part of the ventricular system that's my favorite right here. Tells me it's midbrain. This is your cerebral aqueduct, right? So there's your cerebral aqueduct, and that's my midbrain. Okay, so everybody see where we are? Let's start labeling parts of the basal ganglia. We've got this structure up here. This structure up here is actually closely associated with the ventricular system. If you remember, your lateral ventricles would be coming out around here, and then that's your third ventricle right between the thalamus, comes down to the cerebral aqueduct. So this is just going to be tightly associated with that ventricular system. And these structures bilateral. This is known as the caudate, C-A-U-D-A-T-E, nucleus. Remember, a nucleus is nothing more than gray matter in the CNS. This is one of those islands of gray matter that we talked about previously. Okay. In addition to the caudate, bilaterally, we have this these structures here. Okay, kind of looks like a slice of pie, right? The outermost portion of the slice of pie is located here bilaterally. This is going to be something known as your putamen. P-U-T-A-M-E-N. Putamen is just going to be another part of the basal ganglia that we're going to talk about. And you should know that both the putamen and the caudate nucleus function together, and they together, both the caudate and the putamen have a special name. That means that you're talking about the two of them. That's called the striatum. S-T-R-I-A-T-U-M. So the caudate and the putamen together, because they often function together as a pair, are known as the striatum. Caudate separate, putamen separate, but together they function, and you're going to see how they function together today. They're called the striatum. And then inside here, if we just go more medial to the putamen, the first area that we come upon here on both sides. Okay. This is going to be, I will abbreviate it as GPE. And GPE stands for globus, G-O-B-U-S, pallidus, P-A-L-L-I-D-U-S. And then the E stands for externus. So your GPE is your globus pallidus externus. And the externus here is the one closest to the putamen, the one that's more lateral. And then just inside the GPE, since you have a globus pallidus externus, there's got to be a globus pallidus internus. So this is going to be your GPI. So just medial to the GPE is your GPI, and that's globus pallidus internus. So the most medial of the two globus palliduses. And right now all we're doing is just going through and labeling structures that these are all considered part of the basal ganglia system. Okay, so like I said, the caudate nucleus and the putamen function together as the striatum. This pie slice, the putamen, globus pallidus externus, and globus pallidus internus together constitute one thing that's known as the lentiform nucleus. So all these things together, this is called the lentiform, L-E-N-T-I-F-O-R-M nucleus. Really, that's just the name for the slice of pie. That whole slice of pie is called the lentiform nucleus. So just a lot of new words. Okay, and as I pointed out, this is going to be my thalamus. And then just underneath my thalamus, it's easy to remember, this bilaterally is going to be your subthalamic nucleus. Your subthalamic nucleus is just below your thalamus. Makes sense, right? last part of the basal ganglia we're going to talk about today is actually present in the midbrain, and it's this right here. In that area that I've covered black for a reason. It's known as your substantia nigra. Or black substance. It actually looks black. If you took a midbrain and you did a cross section through it, you could actually see it. Labeled black. Yes? How it is? Are all the what things? Are all the things that you colored in all part of the basal? Yes, everything but the thalamus. Yes. So parts of the basal ganglia, caudate, putamen, those palace, internus palace, externus subthalamic nucleus, and substantia nigra together constitute that group of structures that are considered basal ganglia. So just let me know if I need to move the thing if I zoom in. Is that better, or do you want more than that? Is that good? All right. You're welcome. Okay. Yes, Alice.
When you have like autoimmune diseases like in the basal ganglia, what nucleus does it like have?
Autoimmune disease in the basal ganglia? I don't know that I know. Do you know, Dr. Blair, autoimmune diseases? I've never heard of an autoimmune disease. Encephalitis is swelling of the brain. That's not typically autoimmune, though. Did you have something else to say?
I haven't heard of any autoimmunes that specifically affect the basal ganglia.
Yeah.
Do you
So
need immune deficiency? Is that what it is?
immunodeficiency?
Maybe.
Or the basal ganglia? No. The only thing I can think of that has like a disease. There's something called shoot, it was just there a minute ago.
It's not really.
It's Wilson's disease. Wilson's disease is a disease where you have difficulty with copper metabolism, and it affects the lentiform nucleus specifically, as well as the liver. Those are just two areas. But that's Wilson's. That's something. The only thing that I can think of at the moment that has anything to do with the immunodeficiency or like a nutrient. It's a copper metabolism,
I think it's
probably.
It was listed like today we were talking about immune deficiencies, and like meningitis was listed, but that wasn't as an immunodeficiency. We were just talking about different like, infections. I think.
Yeah, meningitis is going to be more of an infection of the meninges,
I think
like
that's
Dora, arachnoid, and Pia. Okay?
I don't know if that's
Any other questions? So these are all the basal ganglia, and the substantia nigra we're going to talk specifically about because the substantia nigra is actually where something known as dopamine is produced. Dopamine is one of those neurotransmitters that we talked about previously that is neither can be both excitatory and inhibitory. We're going to see that today. Yes, Mike?
So we have the BPE, GPI, and all of that part of the basal ganglia communicating to M1 and all of that.
You're going to see that in a minute. We're going to draw all that out.
But the caudate nucleus doesn't.
Yes, no. So what's going to happen, you're going to see a pathway that the motor planning area, the SMA and PMC, is going to communicate directly with the striatum, both the putamen and the caudate nucleus, and that's going to run through either the direct pathway or the indirect pathway. We're going to draw all that out.
That was me.
Okay. Any other questions? See where we're going? So this, all I've done here is labeled and indicated what is considered the basal ganglia. All right, let's go ahead and move on then. Let's talk about important thing for following the pathways, and that's going to be your neurotransmitters, receptors, and action potential. Okay. So what I've done here is on the left side and the right side, this is going to be my presynaptic neuron, and this is going to be my postsynaptic neuron. Okay, and important for understanding the pathways that we're going to talk about is going to be the color. I'm going to keep my colors very consistent here, and when you see a neuron or when you see something that is colored red, so this first neuron I'm going to be using the color red. Okay. this neuron is going to be a glutamate releasing neuron. In other words, say that as glutamatergic. So a glutamate releasing neuron. Okay, so if you see the red color, that means is glutamate an excitatory or inhibitory neurotransmitter? Does anybody remember?
Excitatory.
Excitatory, yes. The main excitatory neurotransmitter in the CNS is glutamate. So this guy is gonna, what if it releases glutamate? I'm gonna represent my glutamate here as just my neurotransmitter being released, and then binding on that postsynaptic receptor. Again, this is a glutamate that's being released. If this neurotransmitter is excitatory, is it going to depolarize or hyperpolarize the next neuron in sequence? Depolarize, right. So glutamate, when it binds to the receptor on the postsynaptic neuron, is going to cause depolarization of that neuron. Okay, so that means that that neuron is going to fire, right? You're going to have that action potential going all the way down. So what happens is binding of glutamate by the postsynaptic neuron is going to cause an influx of sodium. Let me make it a different color. You're going to have an influx of sodium into the cell, okay, and that sodium is going to cause depolarization of the next neuron in sequence. That makes sense. It's been a while since we looked at this, but that's why we cover a little bit of the action potential stuff in the beginning of the semester just to understand this lecture. Yes?
What does NA do again?
Sodium is going to come into the cell, flood into the cell, causing that cell to become more positive, resulting in action potential happening. Okay? Okay. Questions about that? So as the glutamate binds the receptor, causes opening of sodium channels, causes sodium to come into the cell, that cell becomes more positive, resulting in a depolarization because it crosses threshold. Okay, so remember that sort of the action potential stuff we talked about previously? Okay. All right. So red, glutamate releasing, depolarization happens to the next neuron in sequence. The other color we're going to be consistent with is blue. Okay. Okay, if you see a neuron or neuronal cell body and an axon indicated in a blue color, this is going to be a GABA releasing neuron. GABA. GABA aminobutyric acid. Gamma aminobutyric acid. So that's going to be a gamma releasing neuron. Okay. Was GABA excitatory or inhibitory? Inhibitory. Perfect. So as that GABA releasing neuron releases GABA, that means that that GABA is going to come and bind to the postsynaptic neuronal receptor. And does that cause depolarization or hyperpolarization? Hyperpolarization, right? So it's going to make it harder to fire. So this is hyperpolarization. Okay? So when GABA binds to its receptor, what happens is that potassium channels open and potassium rushes out of the cell, making that cell more negative or less likely to reach threshold. Questions about that? This is important for understanding the pathways, so I'm just taking my time and making sure everybody's got it. So similar to what we had talked about before, glutamate, an excitatory neurotransmitter, causes depolarization of the next neuron in sequence. GABA, inhibitory neurotransmitter, causes hyperpolarization of the next neuron in sequence. All right, moving on down the chain, we've got the last neuron here, color. And I'm just going to make it gray. Okay. And this group. Do you have a question?
Oh, for gray.
You want for gray? Sorry.
We'll work it out.
All right. Okay. The gray color is going to be a dopamine releasing neuron, or a dopaminergic. Dopamine. Okay, that dopamine releasing neuron. Going to be a little bit different. I said previously that dopamine can either be excitatory or inhibitory. Does anyone know how that happens? Happens due to the kind of receptor that's on the next neuron in sequence. Okay, so the postsynaptic neuron. If the postsynaptic neuron has a D1 receptor, okay, something called a D1 receptor, and the dopamine that binds to the D1 receptor is going to cause sodium to flood into the cell, which results in neuronal depolarization. So it's excitatory. Okay, so if dopamine binds to a D1 receptor, which is the receptor on the next neuron in sequence, that's going to be excitatory. So you're going to cause depolarization of that neuron. Okay. And conversely, if dopamine is released and then binds to a receptor that is a D2 receptor, that D2 receptor is going to make dopamine inhibitory, causing the efflux of potassium out of the cell, resulting in hyperpolarization. Of the next neuron in sequence. As we go through the pathways, it's going to be really important to keep in mind the relative concentrations of each of these neurotransmitters in their different spaces. And those concentrations are going to change depending on what's before it, after it, what the disease process is, whether it's a D1 or a D2 receptor. All of those things are really going to make a difference. Okay? Questions before we get into the pathways? Okay, let's go forward. So these are neurotransmitters and receptors. Now we're getting into the first nigrostriatal pathways. Okay, what I've done here is I've just taken, we drew this earlier, so I've taken this picture and I've just cut it in half. So now you're just looking at half of that. You see, so here's going to be my longitudinal fissure right here, so I'm just looking at this half of the cortical region. Okay. And just as a quick review, let's go through and label some structures. Does anybody remember the name of this structure here? That's your caudate. Good. It's your caudate nucleus. There's your caudate. Okay. Everyone knows this is your thalamus. All right. Seen that a bunch of times. What's the thing underneath the thalamus called? Here's your subthalamic nucleus. over here, if we look at the pie slice, we have our GPI, or globus pallidus internus, right? So this out most inner portion of the pie slice, the angle. And then next to that, we have globus pallidus externus, or GPE. And then most lateral, we have putamen. Okay, and then down here, we have this. This is that piece, part of the basal ganglia that was present in the midbrain. This is going to be your substantia nigra. The only other thing I'm going to draw in up here, make it in black, this is that area that's involved in motor planning, or two of the areas that were involved in motor planning. I'm just going to link them both together because they function together. So this area up here in the cortical region is going to represent both your SMA and your PMC, right? Involved in motor planning. Before M1 can fire, these guys have to do something. Questions? Okay, let's go ahead and see what we got here. So, there's my blue. Okay, so if you start in the, think about how to start. Okay, always present when you're at rest, nothing's happening. Oh, first of all, this is the direct pathway. So first we're going to look at the direct pathway. At rest, present at rest, in the globus pallidus internus, there's going to be a neuronal cell body. It's blue. What does it release?
GABA.
GABA. Right. Blue releases GABA. Okay. This globus pallidus internus neuronal cell body is going to make a connection with, and this is always on at rest. Okay. It's going to make a connection with another neuronal cell body in the thalamus that's red. So that releases glutamate. Okay. And this is at rest. So that is going to make a connection up here with your SMA and your PMC, or your motor planning area. Okay. This nucleus, or this, yes, Johnny?
So that's just a normal neuron that's in the thalamus.
This is at rest. At rest, yes, it's just a normal neuron. It's a neuronal cell body that's in the thalamus, and there's a neuronal cell body in the GPI. And granted, this is oversimplified. I'm just trying to get you to see some things. Okay, this is at rest. Okay, this neuron in the GPI is always on. If it's always on, what's going to be released here? GABA. If GABA is released here, is this going to be firing or not firing? Not firing. It's hyperpolarized, right? So at rest, when you don't want any movement, firing is inhibited. You got it? Does that make sense to everybody? This is always on. Okay, that's the first part. Next part. So now you're thinking about moving. Okay, you're thinking about moving, and there's a neuronal cell body that exists, just combination, SMA and PMC. Okay, that neuronal cell body is red, so it's going to release glutamate. Right? So this neuronal cell body sends out axons that are going to contact the striatum. And the striatum, if you remember, is both the caudate and the putamen. So there's an axon that comes out here, synapses with something in the caudate, which is half of the striatum, and it also synapses with something here in the putamen, which is the other half of the striatum. Yes?
Does the red mean anything from the thalamus to the PMC in a sense.
Yep, it means that that's going to be releasing glutamate. Okay, if it depolarizes, it releases glutamate, and that promotes movement up here. Does that make sense? Okay, so red, stimulatory. But right now, at rest, it's inhibited by the neuron coming out of the GPI. Yes, Taylor is.
Once you get into those red pathways, we're not at rest.
Yeah, so what's happening is the SMA and PMC is sending out, it wants to contact the basal ganglia and know, but we're starting to plan a movement, so we want to know where things are and whether or not we're going to move. Okay, so think about motor planning, what has to happen there. Okay, so everybody got it. Okay, so what happens is there's another neuronal cell body here in the caudate that it makes contact with. Okay, and there's also one in the putamen. Okay, and these collectively from the striatum make a connection with that neuronal cell body in the GPI. Everybody got it? All right. Now, coming out of the substantia nigra, we also have some connections to make. So substantia nigra is also going to make a connection here with this neuronal cell body that exists in the putamen, as well as the neuronal cell body that exists in the caudate. Now, are those neural connections? So these are dopamine releasing neurons. So I'll even write dopamine releasing. Are these going to be stimulatory or inhibitory? Stimulatory. What was that? Stimulatory or inhibitory? Did anybody? I heard stimulatory. I think I said stimulatory, but how do we know if it's going to be stimulatory or inhibitory? D1 or D2, right. So the receptor here, if I told you that these receptors were D1 receptors, that means it's going to be stimulatory, right? You're going to cause depolarization. Everybody got it? So let's go through and walk through here. So you have, this is a glutamatergic neuron or glutamate releasing neuron. Okay, so coming down here, you're going to have an increase in glutamate, right? Just by this. Okay, and you're also going to have an increase in dopamine, and that increase is going to be stimulatory. And I'll just put a positive because it's stimulatory. It's stimulatory there, and also it's going to be stimulatory here. So you have a combination. You have dopamine that's going to cause depolarization, and you also have glutamate that's going to cause depolarization of the next neuron in sequence. So it's in contact with a GABAergic neuron. So are you going to get GABA here or no? What do you think? Is there going to be a lot of GABA released here, a little GABA released here, no GABA released here? So these neurons are going to depolarize. If they depolarize, will they release their neurotransmitters? Yes. Okay, so you're going to have a lot of GABA released here. Right? Does everybody see that? I think I lost everybody. Yep. Mm-hmm. Yep. That's why it's going to take some time. But that's okay. So you have the red, glutamate releasing. You told me that glutamate and also a D1 receptor is going to cause depolarization of the next neuron in sequence. So you have a combination of glutamate and dopamine that are going to cause depolarization of the next neurons in sequence. So these are the next neurons in sequence. So that means when these depolarize, they're going to put out their neurotransmitter. Right? And you've got two things here that are additive to cause depolarization of the next neuron in sequence. So there's nothing that's going to hyperpolarize. We're saying we're going to fire no matter what. And then you get a lot of GABA released here after depolarization. Yes?
So you said the combination of the glutamate and the dopamine causes depolarization of the next neuron.
Correct. So glutamate by itself could cause depolarization, and dopamine by itself, if it binds D1, can cause depolarization. So you have a double whammy here, right? You've got both the dopamine in a normal human population. You have both the dopamine and the glutamate causing the release of two things that are going to depolarize the next neuron in sequence, and this is normal. Okay? Yes, Talia? So the only thing that we started here, this is always releasing GABA. So this is always inhibitory when there's no movement. Okay? So it's not releasing GABA until it's stimulated to release GABA or stimulated to release less GABA. And like I said before, you're really concerned about the change in the concentration of neurotransmitter here. Okay? So you have a lot more GABA released because you have glutamate and dopamine. Does that make sense? Go ahead, Taylor Grace. Maybe that'll help.
Okay, so I'm assuming these are GABA, but so are the top two blue ones going to be releasing GABA too, like the ones on the left, before they get to the one at the bottom right?
Okay, so let me point, and that way I can help. Okay, so this guy's going to release glutamate. So thinking about moving causes this to depolarize.
Yes.
Okay, so then you're going to release glutamate. If glutamate is released, you're going to depolarize the next neuron in sequence. Okay.
But aren't those?
They're not. So if you want to think about it that way, if you want to consider these always firing, but they're not always firing. Okay.
Are those 2 points, like neurons too? What are those representing?
Those are representing GABAergic neuron or GABA releasing neurons. So these are
releasing GABA.
these neurons will release GABA to the next neuron in sequence. So the blue. Okay, here, let's go back and look at that picture. Maybe that's where I. Where's that one? The blue one. There we are. Okay, let's go back. So, remember I said it's going to be really important to remember that if you see a red color, it's going to release glutamate. And if you see the blue color, it's going to release GABA.
Okay, so those 2 on the left were
GABA.
that GABA one.
They're blue.
And it's releasing GABA in the next one?
It's okay. Yep. So it's releasing GABA to the next one.
So like is that the third one, the next neuron, where it's releasing GABA?
So these are axons coming off of those 2 neurons that are contacting the next neuron in the sequence. Remember, this guy is always on.
Okay, so that is the next.
That's the next neuron in the sequence. That's receiving the signal. Got it? Do you guys have a question back there I can help with?
She was just answering. I asked another one.
You got that? Yeah. Okay. So yes, this is the next neuron in sequence. Okay. I started by telling you that that's always on. Okay. But now what's happening is you're thinking about moving, turns it off. Does everybody see that? You're thinking about if you think about moving, you're releasing glutamate, and then you have the D1 that's causing this to fire, and you're going to increase the amount of GABA here. If you increase the amount of GABA here, does that mean that this neuron is going to depolarize or hyperpolarize? Hyperpolarize. If you hyperpolarize, then it's going to decrease the amount of GABA here. Right? So if you hyperpolarize, there's going to be less GABA here. Okay? And if there's less GABA here, what happens to the neuron in the thalamus? it fires. Okay? So if it's allowed to fire, it's released from inhibition. It's normally inhibited, but it's released from inhibition. If this neuron fires, then you signal to the motor planning area that it's okay to move, because you're going to release glutamate here, and this is going to result in an increase in movement. Okay, another way to think about that is it's okay to start the movement, or movement initiation is okay. Question? The gray and the red are thinking about movement. No, the motor planning area is about the SMA and the PMC. That's when you're thinking of your motor planning. You're thinking about movement. So what happens is all of these pathways are already connected. Okay, they're already connected. The only thing that's changing is when you think about moving, then you cause the release of a neurotransmitter that's either going to depolarize or hyperpolarize the next neuron in sequence. In the absence of thinking about movement, I think it makes sense if you think that the neuron in GPI is always on. It's always releasing a ton of GABA right here to prevent you from releasing. So, if there's a lot of GABA right here, then I'm not going to be able to move because that next neuron can't be polarized. You're moving. You're going to initiate movement. Right.
At rest, GABA is always on.
This GPI neuron is always on. It's inhibiting neuronal firing in the thalamus to communicate with movement. Yeah.
Those two on the left go down to that one on the right, and it's still releasing GABA, right?
Okay. So it's the relative concentration of GABA. So if I have a little bit of GABA at this base and I get a signal to think about moving, then I'm going to really increase the amount of GABA there. If I really increase the amount of GABA here, that's going to cause the amount of GABA at that position to really go down. There you go. Okay. Those are two different points. So this is where we started. This is the neuronal cell body, and this is the axon. So the action potential travels this way down to the synaptic cleft to the next neuron in sequence, which is the postsynaptic neuron, which is here. Exactly. It's the concentrations of the neurons. Does that make sense? No? Go ahead.
So in the thalamus, when the GABA releasing one is connecting to the glutamate releasing one, if the concentration was increased for GABA, then it wouldn't fire the...
Yes. And that's normal. That's normal movement. Okay, in the normal situation, but in the absence of any movement, there's always GABA. There's always a bunch of GABA there to prevent that neuron from firing because you're not wanting to move yet. It's important. Okay. Any other questions? Does it make sense? I think when I put the Parkinson's, hopefully it'll make a little bit more sense. But let's go ahead and take a 10-minute break.